Alright guys, here's part 4 for chapter 3, going through protein synthesis now. We've talked about this a few times already. We talked about it when we did proteins, and we talked about it at the beginning of this chapter when we talked about ribosomes and that process of how proteins are formed. I'm just going to go a little bit more specific into it here so you guys can understand how the whole process works. So some of it can be review. Again, this is going back to biology stuff, so hopefully you remember a little bit. At least some of it's a little familiar, but this is the process here. So you may remember the terms transcription and translation. That is how protein synthesis occurs, and you start with DNA and you end up with protein. So we're going to go through how that whole process works. And this happens in all cells, eukaryotic and prokaryotic cells. If you remember from biology, eukaryotic cells are cells that have a nucleus. Prokaryotic cells don't have a nucleus. And basically prokaryotic cells are bacteria and other organisms called archaea, which are not as common. They live in extreme areas, but bacteria is mostly prokaryotic cells. Eukaryotic cells are everything else, us and plants and other living things. So the DNA in all cells, though, goes through a process of protein synthesis. So in prokaryotic cells, you have the DNA just inside the cytoplasm. There's no nucleus around it. It doesn't have that nuclear envelope. It's not contained. So it's just in the cytoplasm, and there's one single long DNA strand all intertwined with itself in that cytoplasm. And that intertwined DNA, that single strand, can come together and form what's called a chromosome, which is a more uniform shape of all that DNA strand all packed together. And then in that chromosome, the sequence of DNA that codes for a protein is a gene, and that determines a trait. So that protein that's formed can create some kind of trait, whether it's a tissue or an organ or some type of hormone or enzyme or whatever in your body. It's going to affect some type of function in your body in some way. So the chromosome structure, you have these DNA molecules, and like I said, it's a long strand of that double helix, and they're actually really strong and really long. I mean, if you had to think about it, kind of an example is if you had a rope and you laid it out and it was about 300 meters long, which is longer than 3 football fields. If you tried to stuff that rope into your backpack, that's what it's like trying to put that DNA inside a molecule or inside the nucleus of a molecule. So that's basically that's kind of what it's like, and it's really folded up and tightly packed to fit into there. Chromatin is granular material visible within the nucleus. So we talked about chromatin a little bit when we went through all the organelles. All this is chromatin, and that's more tightly packed together, and you have a form eventually of like the chromosome. So this is all the DNA being tightly packed together, making that chromatin. Proteins are made up of amino acids. We've talked about that multiple times, so you should know that now. And we talked about enzymes when we talked about proteins. So enzymes are proteins, remember, and they're catalysts that speed up reactions in your body. So that formation of those proteins is established inside your cell, in each individual cell. So that's what we're talking about today. So the process, in order for that to happen, has to go through transcription and translation. So transcription, when you transcribe something, it's like writing something down, or it's copying something down from something else. So if you had a piece of paper and you were taking information from that piece of paper and writing it down on another piece of paper, that would be transcribing it. So basically that's what's happening in the process of transcription in your cell. You have DNA, you start with DNA, and then it is forming a strand of RNA, which is a complement to the DNA so that it can send its information out of the nucleus. And that process is transcription. Once it gets out of the nucleus and gets to the ribosome, then it's going to go through translation, which is where you're actually forming the proteins. In translation, just like when you translate a language, you take one language and you turn it into another language, and that's what protein synthesis is doing with that RNA. It's taking the information from the RNA and creating proteins out of it. So here's transcription. You have that DNA strand here, and it's going to start coming apart and creating a complementary strand of RNA, which would go together. So we talked about these nucleotides last chapter and how they match together. G's goes with C's, and A's go with T's in DNA, but now you're making a new RNA strand, and you have U's in RNA instead of T's. So A with U, G with C, A with U, A with U. So that's how it's forming that RNA. And this is messenger RNA. Remember we talked about that and when we talked about nucleotides as well. So you get that messenger RNA, which is this one here, and now that messenger RNA can go out into the cytoplasm and go find some ribosomes so it can go through translation to make proteins. This is a little schematic, a little animation to show you kind of how the process works. I'll let you go ahead and look at that on your own, but let's talk about what happens after transcription. So again, you have that mRNA. Remember, the DNA is contained in the nucleus. It's not able to come out of the nucleus. It stays in there, but in order to send its information out of the nucleus, it makes that mRNA so it can go out. See, it's coming out of that nuclear envelope, and it's coming out into the cytoplasm so that it can go find some ribosomes, or that rRNA. Once it does that, that mRNA's got codons on it. Those codons are 3 nucleotide sequences, which are basically a code that tells the tRNA which amino acids to use. So this code right here, AUG, is a code for a specific amino acid. AAC is a different amino acid. And you put those together, and that makes a protein when you put amino acids together. So the language of mRNA is the genetic code, and that's what's being translated when you're going through translation with the mRNA and tRNA, which join each other at the rRNA. So here's the little schematic about it. So here's the mRNA here. This gray substance here is the rRNA, and then these blue boxes are the tRNA, and you see how they have the 3 nucleotide code also, and that's the complement to the 3 nucleotide code on the mRNA. So GAU goes with CUA, and then you got CCC here. This GGG is going to move in here and then bring this amino acid with it. So these are all amino acids here lined up, and they're forming a protein. So begins with mRNA with translation. Each codon of the mRNA has a complement on the tRNA, which meets at the ribosome, and that forms the protein that codes for whatever tissue, enzyme, organ that protein is being formed for. So that 3 nucleotide code on the tRNA is called an anticodon. So on the mRNA, it's called a codon. On the tRNA, it's called an anticodon, because it's the opposite. It's a complement to it. Those coming together is what decides which amino acid is going to be added to the protein. So in case the tRNA molecule for methionine, here's methionine. UAC is the anticodon for methionine. The codon for methionine is AUG. Then you have valine coming in also here, which is an amino acid. that anticodon is CAG, and the codon is GUC. So that's put those two together. That's a peptide bond, and that even if you have two amino acids, that's a protein. All right, here's another schematic showing you the whole process: transcription, RNA coming out, mRNA coming out here to the rRNA, and the tRNA is coming in, and it's forming that bond in that long chain of amino acids. Here's all the codons for the different amino acids and how they go together. This is all determined by the DNA sequence and how those nucleotides are lined up with each other and which ones are there. So here's another picture of how the whole process works. This is mRNA moving in here, and here it is with the ribosome or the rRNA, and here's tRNA bringing in the amino acids and building up this protein chain. Okay, again, another way of looking at it. Occasionally, there's a change that occurs in the sequence that can cause a mutation. So a DNA sequence can have an alteration sometimes. Mutations are not that common though. There's a 1 in 100,000 chance of a mutation occurring in a DNA sequence. So fairly rare, but it does happen, and when it does, abnormalities or things that are not quite the norm occur. Like this right here, you see the frog's got extra legs. It does happen, but not very common. This alligator's only got his head is white, the rest of his body's green. So that's from a mutation. So there's different types of mutations too, though. There's a point mutation, which means in a point mutation, one single nucleotide has been affected, and it's been substituted for something else. So here in this example, you got AUG-UAC, which is methionine and tyronine, but there's a mutation. Instead of the C, it was replaced or substituted by a G, and that created a stop sequence, which makes that stop sequence ends the production of that protein at that point. So it would stop that protein production early, which could affect the growth of something. So that's a point mutation. One substitution of one nucleotide is a point mutation. So one way to think about that is it's at one point in the DNA sequence that something changed. Now, a frame shift mutation is different. In this case, you can have insertions and you can have deletions. You could have a nucleotide added in a random place, or you could have one that was removed in a random place. So, like the example here I have at the bottom, original here, the fat cat ate the wee rat. Well, if you take away this T and shift everything to the left, then you end up with this sentence, which obviously doesn't really make sense, but that's what happens in the genetic code too. That sequence doesn't really make sense, so it changes the way that whatever protein is being formed is produced. So it affects the way things look, which is called the phenotype, if you remember that. So the genetic code is called the genotype, and then changes in the genetic code or the genotype can cause effects and changes to the phenotype, which is the way things look. So here's an example of a frame shift mutation, and it can change the whole sequence of amino acids just because of one shift, one change. Significance of mutations: they can have no effect, they can have a little effect, and they can have abnormal effects. Sometimes they can be beneficial as well. They can help you adapt to different environments and things like that. So there are positives and negatives to it, but again, it's not something that commonly happens on a regular basis. So they occur, the changing sequence of nitrogen bases in the order of the nucleotides, which affects proteins production. And This has been part 4, and we are now done with chapter 3.